In this video, we will be covering point mutations. Let's take a look at some examples. Here we have a coding strand of DNA in the 5' to 3' orientation. When the template strand is transcribed by RNA polymerase, we will get the following 5' to 3' mRNA sequence where the thymine bases in the above DNA are replaced by uracil. Now we can use the coding dictionary to determine the amino acid chain this protein codes for. First, we need to find the start codon, AUG, which codes for the amino acid methionine. The next codon is GUC, so we get valine. AGC codes for serine, AAG codes for lysine, and UAA is a stop codon, indicating translation ends here. Let's see how this protein is impacted by some point mutations. At the top here is our original DNA coding strand, the transcribed mRNA, and our amino acid sequence. And at the bottom, we have the same DNA sequence, except with a single base substitution. When we convert the coding strand to mRNA, we get the exact same sequence as we had originally, except the cytosine in the third codon has been replaced by adenine. Therefore, this is a transversion mutation. The first two amino acids are the same, as the codons are identical. However, instead of serine, our third codon codes for arginine. The last two codons are the same, so we get lysine and stop. When a single base substitution causes a codon to code for a different amino acid, this is called a missense point mutation. If the original DNA sequence was a beta hemoglobin gene, a missense mutation could result in a misshapen hemoglobin protein that causes the disease sickle cell anemia. Let's look at another mutation. Here we see that an adenine in the original DNA sequence has been replaced by thymine, which once transcribed becomes a uracil, indicating another transversion mutation has occurred. The first three codons translate to the same amino acids, methionine, valine, and serine. However, where we code for lysine in the original sequence, there is now a stop codon, UAG. As a result, the newly synthesized protein is only 3 amino acids long. A single base substitution that causes an amino acid codon to be read as a stop codon is called a nonsense mutation. If the original DNA sequence encoded for a functional CFTR protein, a nonsense mutation within it could cause an individual to suffer from cystic fibrosis. However, not all mutations have negative effects. In this sequence, a transition has occurred where a cytosine has been replaced with a thymine. This creates an mRNA codon that now reads as GUU instead of GUC. However, the amino acid sequence we get from translation is the same. This is because multiple codons can code for the same amino acid. A point mutation that does not change the amino acid sequence is called a silent mutation. In this last example, we will examine the impact of a transition point mutation within the DNA region that is transcribed into our stop codon. Here, thymine has been replaced by cytosine. As a result, instead of reading as UAA, the stop codon now reads as CAA, which codes for glutamine. With no stop codon in sight, translation will continue, giving us another serine and a threonine. The resulting protein is 3 amino acids longer than the original. A point mutation that, when transcribed, changes the stop codon into an amino acid codon is called a read-through mutation.